Hi, I'm Dr. Lawrence Cooper at the MD Anderson Cancer Center, and I want to tell you a little bit more about our program in applied cell therapy. With me today is Dr. Kirsten Switzer, an outstanding biologist who's working to help us understand the potency, in other words, the ability of a T cell, a type of immune cell, to target a tumor cell. Kirsten, what's going on in your work?
So we use the mouse as our main animal model right now, most versatile animal model, and we engraft human lymphoma and then treat them with our different T cells that we create in the lab.
Wow, so you can put human T cells and human lymphoma cells into a mouse.
Right, these mice have no immune system, so they accept the human cells without problems.
Fascinating. So when you put the T cells and the tumor cells together, what happens?
Well, so far we've had a lot of success in mice, seeing that the T cells decrease tumor in mice. And
So the T cells can target the tumor cells?
yes.
Huh. And how do they do that? Are these just anybody's T cells out there, or are these special T cells?
These are T cells specific for CD19, which is found on most B-cell lymphoma.
Huh.
So,
So you've made the T cells target the 19 molecule on the lymphoma cells.
right,
And once the T cells see the lymphoma cells blow up and die.
right.
So how do you know the lymphoma's gone? How do you know the mice are cured?
So we monitor tumor burden and tumor reduction via bioluminescence imaging, which is very
Kind of sensitive?
very sensitive, and so to do that we label our tumor cells and our T cells with different bioluminescent labels, and then we are able to image the
Wow.
T cells or the tumor cells.
So these bioluminescent labels that you're putting in there, so how does that work? Just tell us a little bit more about what bioluminescence is.
So bioluminescence occurs in nature in different organisms naturally, so they've used that and put those molecules and tag cells with them, and then you use a substrate for that enzyme, and its chemical reaction creates the light.
Wow.
Basically.
So the tumor and the T cells are using this bioluminescent property to make light.
Right.
And then you measure the light, and essentially you know if the light goes away, the tumor's gone away.
Correct. Right. And you can quantify it, so you can do all kinds of statistical analyses, and it's very
Terrific.
very useful.
Terrific. Well, congratulations on your work.
Thank you.
So Kirsten's really helping us, because with these types of techniques, bioluminescent imaging, where she can understand the ability of T cells to navigate to tumor and to kill tumor, she can help essentially with the question, does it work? Do these human T cells in the mouse get rid of lymphoma? Do these human T cells in the mouse target CD19 on the lymphoma? And when they do, as she so nicely showed, my job then is to put that into the clinic. So we have ongoing clinical trials right now where we're taking these types of T cells and we're putting them into patients with the question of whether the human T cells in the human can get rid of their CD19 positive B-cell lymphoma. Thank you very much.